Hi everyone, welcome back. There are many things I want to cover in today's video. Let's list them out one by one. First, it's been some time since I last talked about anything biotech and I'm returning back to my roots. Today, I'm making a comparison between the old and the new gene therapeutic methods and also explain why the old method does not work using Sarepta therapeutics as an example. In addition, I spend a lot of time and effort trying to make my videos as educational and beneficial for all of you channel supporters. And on that note, there's a major announcement that I'd like to make at the end, so stay tuned. But before that, let's roll that intro. I've been teaching biology since 2004. On this channel, I hope to simplify and explain the science behind the companies that's driving the genomics revolution, one video at a time. One of the first gene therapies involved treating a medical condition, SCID, which was initiated back in the year 2000. Patients suffering from this genetic disease did not have any functional immune system and have to live in sterile rooms in hospitals. Any contact with microbes will mean inevitable death, as they succumb completely to the infection. The reason behind the disease is because of small mutations occurring on a gene on chromosome X, which codes for a cell surface receptor on white blood cells responsible for binding to cytokines. Cytokines are a very diverse group of chemicals that immune cells produce as a way of communicating with each other. So if immune cells cannot communicate, they cannot therefore produce the vital microbial fighting molecules such as antibodies. The idea the scientists had was to use a virus that can bring the corrected version of the gene into these immune cells. Retroviruses are able to do that, and one such example is HIV. Since normally, as part of their reproductive cycle, they have a gene called integrase that aids in the insertion of the viral genome into humans. The scientists will then take out viral genes that are disease-causing and replace it with the corrected version the patient needs, and then infect the patients with these recombinant viruses. However, within 2 years of receiving gene therapy, a French boy that's enrolled in the clinical trial developed leukemia. This is a type of white blood cell cancer. Aiyo! Not only that, this was immediately followed by yet another boy enrolled in the same trial, developing the same cancer. So the question is, what caused it? One potential reason was that the retroviruses do not insert the corrected gene in the exact original position. Instead, the insertion is completely random, which in turn could disrupt the normal functioning of another gene by chance. This is known as insertional mutagenesis. Since this particular clone of cells have the corrected version after gene therapy, it could be selected for and went through multiple cell divisions, which can lead to further accumulations of cancer-related gene mutations. At the same time, there was another gene therapy clinical trial that is targeting another disease where a boy by the name of Jesse Gelsinger died. In light of these unfortunate outcomes, all 27 gene therapy clinical trials are ordered to shut down. And this is the reason why I'm happy that my favorite gene editing company, Bibs Therapeutics, is taking time to submit their first IND. As an investor, as much as we want things to go, we are dealing with human lives here. Better to ascertain the safety right from the start so that the trials can proceed successfully. After the horrendous outcomes, scientists designed workarounds to improve on the old gene therapies. The thinking was that if integration is going to cause insertional mutagenesis, then what about not doing that at all? Instead, just find another virus that can bring the corrected gene into the cell will do. That is what Sarepta Therapeutics did, using AAV targeting a devastating disease known as Duchenne muscular dystrophy. This disease affects mainly boys and it results in the slow but eventual death of muscle cells, and they typically don't live past 20 years old. However, the clinical trials were dismal, showing no improvement in a statistically clear way. The explanation given was that there was unlucky randomization of sicker kids to the treatment group. I have a big problem with that conclusion. If the treatment works, it should work. There should not be any need to look at the nitty-gritty details like that. Let me try to explain why is it that Sarepta's strategy does not work. In the case of Sarepta Therapeutics, the designed gene therapy is done in a three-pronged approach manner, comprising of a virus that carries the genetic payload, which needs a switch in front of that so that gene expression can turn on. There are a few problems with this. First, in adapting the treatment for Duchenne muscular dystrophy, the corrected gene is controlled by a promoter known as MHCK7. This is a 770 base pair promoter that is not original to the dystrophin gene. A promoter functions as a switch for genes after it to turn on or off. An original promoter is important because it will respond in the way that is intended in the normal environment. And the way to do that includes sequences far upstream of a promoter known as distal control elements, which is needed by activators to bind to form a transcription initiation complex that then drives gene expression. In this situation, the promoter included lacks these sequences. The TIC that forms is not the original, potentially affecting gene expression once it is introduced into the muscle cells. In addition, the genetic construct lacks important chromosomal features known as centromeres and telomeres, both of which are crucial for long-term sustaining presence in the cell. Otherwise, free-floating DNA without these structures are easily degraded, making the whole exercise a waste of time. In contrast, the reason why I like Beam Therapeutics so much is that they are not trying to introduce the corrected gene sequences into the target cells. The old method of gene therapy has been shown not to work since day one, back in the year 2000. Instead, they focus on the genetic sequences that's already present in the cell and base editing it to what it should have been. Everything else is left in their natural state. This approach by Beam Therapeutics will likely increase the chances of success. I think an Elon Musk quote is very apt here.
The best part is no part. The best process is no process.
To add on, Sarepta Therapeutics have recently started to transit to gene editing. This goes to show that the advantages of the technology cannot be ignored in light of their poor results obtained from the old gene editing methods. So the next question is, will they be a viable contender in the gene editing space? My short answer is no. Because they have to play catch up, they have no intellectual property, which means they have no moat. And on top of that, they have to pay for licensing fees unless they invent their way out. And this will require both time as well as a scientifically brilliant team. And all of you know who my vote goes to in this arena. I'd like to take time to thank all of you channel supporters for continuing to share, comment, even like and subscribe. I don't push for this much in my videos because I believe if you are moved, you're gonna do it wholeheartedly. Even then, the growth of this channel has been steady and it's a surprise to me. And for that, I'm indebted to you. In the past few months, some of you have indicated that you want to show support beyond the normal ways. I've been thinking about Patreon, but I'm not convinced about it for two reasons. Firstly, it has a censorious history to it, and that extends to this platform as well. Right now, I'm towing the lines very carefully, and to that end, I'm not able to express what I truly feel. Second, I feel that this is a one-way street where you shell out some dough for me to make more bread. This is just too transactional. Like it or not, you are now part of my life and me yours. And bringing money to the table just spoils everything, which is why I've decided to set up camp on Odyssey. Because they have a much freer spirit and also along the way, you can benefit as well. It is a win-win. For example, if you follow and watch videos on my channel, you will gain some LBC, so will I. Again, win-win. I will put the links in the description below for the registration, as well as my channel URL. And just so you know, there are no costs involved. In the future, I'll talk more about it. And in fact, I'll likely make content that's a little bit more risque exclusively over there. Otherwise, it's business as usual on this channel where you stay up to this point because you've been also awesome, and I'm Benjamin Yang. See you in the next video.